Today we are talk about DNA fingerprinting techniques. The DNA fingerprinting techniques observed by Alex Jeffrey in 1984. First, we talk about the definition. DNA fingerprinting is a technique to obtaining the images of DNA of man on an X-ray film. DNA fingerprinting it is used for identification of individual. DNA fingerprinting, otherwise called DNA profiling or molecular profiling. In previous days, thumb impression or fingerprint on paper to identify the individual person. In DNA fingerprinting, by using the X-ray film to identify the individuals. So the thumb impression is unique and permanent for each and every person. It is also like that DNA samples is also unique and permanent for each and every person. In DNA fingerprinting, we use the satellite DNA. Satellite DNA is otherwise called mini satellite DNA. It is nothing but non-coding region of eukaryotic genes. It is having 10 to 40 base pair of nucleotide. It is high content of GC rich, guanine and cytosine. The mini satellite to identify the person. It is where the mini satellites vary from individuals to individual. So it is called VNTR. VNTR represent variable number of tandem repeats. It is a VNTR represent mark. It is for used for marker key for identification of DNA fingerprinting. Then methodology. The DNA fingerprinting have a 9 steps for methodology. First, isolation of cell DNA. Second, restriction digestion. Third, gel electrophoresis. And fourth, southern plotting and packing. And fifth one, selection of probe and filter hybridization. And autoradiography, analysis of print pattern. This is the diagrammatic representation of DNA fingerprinting methodology. In DNA fingerprinting, first isolation of cell DNA. Isolation of cell DNA to collect this DNA from the crane cells and crane cells. It is used to the source DNA and the suspected DNA. The crime sense rape victim is a source DNA and suspected person 1, 2, 3 or XY ABC. Then restriction digestion. Restriction digestion, the DNA is digested into the small fragment. Restriction enzyme to act as a molecular scissor to cut the DNA into DNA fragment. Then the next step is PCR aggregation. PCR aggregation is polymerase chain reaction. It is used the small amount of DNA is copy into the several copies by using the PCR techniques, by using thermal cycler. Then gel electrophoresis technique. Gel electrophoresis technique by using agar gel electrophoresis. Then the electrophoresis technique to run the source DNA along with suspected DNA X and ABC to run the based on the mini satellite DNA length. Then fifth step, southern plotting and packing. After running the electrophoresis to collect the agar gel, Then placed it nitrocellulose membrane. The nitrocellulose membrane or filter paper placed it over the gel. So the gel DNA fragments is transferred into the filter paper. After filter paper having the DNA bands, then the filter paper dried with high temperature. It is called backing. Then selection of probe. Probe it is used for marker or test. So in India, Dr. Lalji Singh worked as in Center for Cell and Molecular Biology. He developed the DNA probe from the poisonous snake, banded dried. The probe name is PKM probe. Then filter hybridization. Then filter paper, the nitrocellulose membrane filter paper placed on alkali solution to denature the double stranded DNA. So the double stranded DNA denature and bind with and to secondly add with hybridization solution. So the hybridization solution contain the radio labeled probe. Then the probe DNA bind to appropriate mini satellite to form a double stranded. So wash alkali solution to for denature the DNA and probe DNA bind to and formation of double DNA or double stranded DNA. Then autoradiography. This technique, the filter paper or nitrocellulose paper washed with wash solution for unbound probe to wash out. Then the nitrocellulose filter paper placed on over the X-ray film for 3 hours. So the nitrocellulose filter paper, the probe emit the dark spots on the X-ray film. So this is a X-ray film. So the probe, radio labeled probe, emit and formation of double stranded DNA. Then analysis of fingerprint pattern. For example, here the rape victim, the source DNA. The source DNA X, suspected DNA ABC, a person, 1, 2, 3. The source DNA perfectly matched with the suspected DNA. So if match is 100%, the individual responsible for the victim. For example, here suspected X, victim X and suspected person A perfectly matched. So A is there. So A suspected person is the responsible for the rape victim. So the DNA fingerprinting techniques used in to settle the disputed parentage and to settle the murder cases and to trace the date of murder and distinguish the rapist. For example, the date of murder in death of, for example, if a person death after 1 hour, the flies or the insect infest the dead body within an hour. The flies having the developed life stages is eggs, maggots, and adult fly. For example, to run the electrophoresis gel, the X-ray film, victim and the flies band. If a death in the same day, the flies band, no band, so the negative. The day before the death, the flies have positive band or DNA band is present. So based on the DNA pan or DNA print the data matter. So DNA fingerprinting play a vital role in disputed parentage, murder or rapist. Uh, that's all about the DNA fingerprinting vital role in forensic science. Thank you for watching this video. Thank you.